The Earth's arable land continues to diminish. The reasons include the escalating drying of the Earth due to global warming and the salination of farmlands caused by the excessive pumping of groundwater. As a result, we will reach a crisis situation, despite our efforts to produce enough food to feed the skyrocketing population of the world. To solve this problem, DREB is continually conducting research into crops that can be cultivated under extremely dry or highly saline or other harsh environmental conditions. One fruit of these efforts is the discovery of a group of genes that allows plants to withstand adverse conditions encountered during cultivation as the environment deteriorates. When the cells of living organisms encounter extreme dryness, high salinity, and low temperatures, they lose water and eventually die. A stress-resistant gene is needed in order to avoid this. Just how does a stress-resistant gene function? When stress occurs, certain types of genes are able to selectively activate target genes that confer stress tolerance to plants. For example, they are able to deliver commands to activate genes whose function is to raise the sugar content with the cells and thus prevent cells and tissues from being damaged by water loss and other conditions. DREB has succeeded in isolating and identifying the genes and has named them DREB genes. DREB has also succeeded in isolating and identifying stress-inducible promoters which were combined with the DREB genes and introduced into model plants such as Arabidopsis and rice. These transgenic plants were evaluated under greenhouse conditions for tolerance to drought, salinity, and low temperature. As a result, the transgenic plants carrying DREB genes showed tolerance to drought, high salinity, and low temperature. Currently, DREB has been conducting collaborative farm-level research with 5 international research institutes around the world in order to develop stress-tolerant crops such as rice, wheat, and various beans. The agricultural community in developing regions is anxiously awaiting the birth of a novel crop variety that can achieve greater agricultural productivity in these environmentally degraded marginal lands.